Next up we have Tanya Napolitano, who'll be presenting on taking a bite out of pain, characterising non-addictive pain therapies from Venom Peptides.
Hello, everyone. I'm just getting my PowerPoint up. It's not letting me share the power. Okay, I got it. Thank God.
Yep, there we go.
Okay.
Yeah, you're all set.
Okay, hello everyone. My name is Tanya Napolitano. I'm from the City University of New York Graduate Center, and I'm in the laboratory of Mandy Holford as a PhD candidate. And today the title of my talk is Taking a Bite Out of Pain, try to make it a little spooky, of characterizing non-addictive pain therapies from Venom Peptides. And we started this project to create a validated Venom Peptide functional activity pipeline to identify potential therapeutic candidates. So snails are not really what comes to mind when you think of venomous animals, but here's a video to convince the non-believers. So in this video, you see that the snail has its shell buried in the sand, it's hiding itself. And then this orange piece of tissue is called the proboscis, and at the very tip, it has a siphon that's sort of like its nose that smells out its prey, which is this poor fish. And then it also has a harpoon at the very tip that injects the venom into its prey. So as you see, once the venom was injected, it took maybe a second or less for the fish to get completely paralyzed, where a snail can engulf a fish whole. So as you can see, the venom is very fast acting, very potent, and this is what you want for new drugs. So also venom doesn't really seem like an likely source of therapeutics. However, these are 6 commercially available drugs approved by the FDA and on the market. I'll start with Captotin. It was derived from the venom of the Brazilian pit viper, and it's an ACE inhibitor. It's used to lower the blood pressure, as well as Angiomax and Intinegrin and Agriostat. These also are blood thinners. Then we have Exenatide that was derived from the venom of the Gila monster, and it's used to treat type 2 diabetes. And lastly, we have Prlalt that was derived from the venom of the cone snail, and it's a non-addictive analgesic that's used to treat pain and chronic pain in HIV and cancer patients. So how does Prlalt work? So first, there's a site of injury or pain, and then the Prlalt is administered through a spinal tap. And then once Prlalt enters the spinal fluid, it targets N-type calcium channels in the nerve cells and stops the pain signals from propagating, and then the pain is reduced. There's also pros and cons to Prlalt. So the pro is it's 100 times stronger than morphine, so that's definitely a pro, and it's not addictive. But the con is you need a spinal tap to administer this because it's too big to cross the blood-brain barrier. For example, by administering it orally or through IV. So our lab also does work on different ways to administer these peptide drugs, but it's overall a great drug for pain. So snail venom peptides are a viable source for pain therapeutics. This chart highlights several cone snail toxins that are currently under development by various drug companies. I already talked about the first FDA-approved drug from a cone snail, Prlalt, which was approved in 2004 by the FDA. However, you can see there's a lot of pharmaceutical interest in these snails because of how potent and fast-acting their peptides are. But in the Holford lab, however, we study the Terebridae, which is a close family to the cone snail. And in one of our studies, we found that a peptide TV1 that was from Terebridae, it's any peptide that comes from the Terebridae, we call it a teratoxin. So this teratoxin TV1 elicits noxious heat avoidance in Drosophila flies once administered. So this shows us that Terebrid peptides also have an analgesic property that are not connected to the opioid receptor. And also, these venomous animals, such as the Terebridae, have evolved their venom over thousands of years to rapidly and efficiently disrupt macromolecule functions in their prey. So in one venom gland, you can get 50 to 200 different venom peptides based on which species of Terebridae you're dissecting. And these peptides have been found to act on a number of relevant targets, such as drug targets, such as G protein coupled receptors, ion channels, enzymes, proteins, and receptors. So that's why there's so much pharmaceutical interest. So we thought, how can we use computational and experimental strategies to study these peptides and get more therapeutic leads out quickly? So that's where we worked with our collaborators, Dr. Nick Tadanetti's lab at Columbia University, also in New York City, and we're doing a Venom-seq pipeline. So in this pipeline, first we have to synthesize pure venomic peptides, or teratoxins that were found in our Terebridae transcriptome database. So we synthesize those pure venomic peptides. We incubate them with IMR32 cells, which are neuroblastoma cells. And then after some time, we read their mRNA we sequence their mRNA by plate-seq. And then we use two different bioinformatic algorithms in tandem to generate expression profiles. So we use DE-seq2, which is an algorithm for comparing differential expression in two cell populations. So in this case, it would be the untreated versus the treated cells, and tells us which genes are differentially expressed and how much they are, whether they're upregulated or downregulated. And then in tandem with Viper uses network data to tell us which transcription modules are being differentially regulated. So instead of genes, we're now looking for modules which are controlled by transcription factors. And together, they recover accuracy and improve statistical power when generating these expression profiles. And now these expression profiles is the measurement of activity or expression of thousands of genes at once, creating a global picture of cellular function. So then we can, once we get our own expression profiles from this Venom-seq pipeline, we can compare it to open source data such as Link's profiles, which has over 1.3 mechanism of actions of FDA approved drugs that are already known, and possibly make hypotheses to how our pure venomic peptides are working. So for our Venom-seq results, we tested 11 different peptides of our own, as well as the last two are controls that we bought that we know exactly what they do. And next, we're focusing on how many gene perturbations each peptide gave to the cell after doing these computational studies. So for Cy3-crotamine, one of our controls, we found almost 1,300 gene perturbations. And then only one of our peptides gave enough gene perturbations for us to study it, create a expression profile, and compare it to OpenLink's database. So I'm also just showing you the sequence of them. And so that was our results from Venom-seq. And then we went to the Link's open source data. So this is the GeoLinks data. It's also called the connectivity map by the Broad Institute. It's clue-io. That's the website. That's how you get to the website. And it has over 1.3 million profiles and tools for analysis of expression profiles and just the way different drugs interact with human cells and their mechanism of action. So for the different parameters that they have, I did not click gene knockdown or gene overexpression because that was not done in our Venom-seq experiment. And then as well for cells, they did not have IMR32 cells, so I'm looking at the summary cell line. However, just a little more information for the summary cell line. It's not just the mean or the median. The authors instead go with the 67th quantile to make their summary score because it compensates for some of the biases that the mean or the median might have. So these are heat maps of Cy3-crotamine and MKI 8.7 compared to the publicly available links database. So as you can see for the cell ID, I'm only really looking at the summary highlighted in green, but it's giving us a median tau score. So it's averaging all of these other cell lines. And then it's giving, we do know what Cy3-crotamine does. It was a control. So we looked first to see, does the control match up? So we know for Cy3-crotamine, it's a potassium channel blocker. So that gave us a median tau score of 95. So anything above 90 is significant. We also know it's a calcium channel blocker, and it gave us a median tau score of 97. And we also know it's an antibiotic that disrupts Golgi function. And so this all showed up. So that was perfect. Next, we studied our MKI, and the summary of it gave us only 4 drug or gene name associations to our MKI 8.7. But because we don't want the median tau scores, we wanted to only focus on the summary cell line. We removed those other cells, and now you see just tau score and not median tau score, and we're able to better get a better understanding of how MKI 8.7 works. But still for the control, we still see potassium channel blocker, calcium channel blocker, and antibiotic that disrupts Golgi function. So that's great. And now for MKI 8.7, we found 7 drug or gene names that were strongly associated with MKI 8.7 based off of its expression profile in the IMR32 cells. So one of the drugs that was very interesting to us that it matched so well to was thiosparagine. So thiosparagine is an ATP inhibitor, and that's known to be a good drug target for cancer, muscle relaxants, heart failure, and ulcers. Calcium channels, obviously, for are associated. Blockers are associated with relieving pain, and then it's also a protein kinase C activator, and that's involved in many downstream events in the cell, like mediating immune responses, regulating transcription, cell growth, learning memory, etc. So the next part, now we've done most of the computational work, is to pick several cell-based assays to confirm that MKI's activity that was predicted in Venom-seq is actually true. So we're going to send our compound out to test for PKC, non-selective ATPase activity for both sodium and potassium channels, as well as ATPase calcium channels, and we're going to send one out for Flipper to measure calcium channels. So as for the summary, Peptide Therapeutics 2020 and beyond. So bioactive venom clearly has immense therapeutic potential. However, there's a need to more efficiently screen and characterize these peptides to discover pharmaceutical leads more quickly. And teratoxins are an uncharted territory for finding new therapeutics that can ultimately overcome molecular and pharmacokinetical obstacles in drug development, such as the opioid crisis and things like that. So this project will allow us to create a validated teratoxin functional activity pipeline to identify potential therapeutic candidates. And lastly, I just like to thank the Holford Lab, my mentor, Mandy Holford, as well as all my teammates as shown in the picture, and of course our collaborators at Columbia University and BU Amsterdam.
Thank you.
Any questions?
Thank you so much, Tanya. That was really interesting. We've got a couple of questions. I'll hand you over to Rohit for those.
Sure.
So what would be the selected function of the analgesic peptides that are analgesic for mammals in the snail venoms?
So we're looking mostly for calcium channel blockers or trip channels, anything like that. We're also just looking, just in general, if our peptides can do anything. So maybe it might not be pain related, but if it has any sort of purpose, we're not just going to throw it away. But calcium channel blockers is mostly what we're looking for.
Correct. And is this antinociceptive effect mediated by homologous receptors to the ones affected in the snail's prey and predators?
Sorry, what?
Is this antinociceptive effect mediated by homologous receptors to the ones affected in snails' prey and predators?
Sorry, I don't understand your question.
So is the effect that this antinociceptive effect, this pain-reducing effect, is it produced by the same mechanism as the prey and predators, the effect it has on the
Yes. So they act on calcium channels in their prey as well as, and when you use it on humans, it acts on calcium channels. For example, Prlalt, that's the only one that I can give an example of because that's the only one that's been studied in various animals that I know of in detail.
Okay, yeah. And next question is, what is your basis in selecting which venoms to synthesize for testing?
That's a good question. We usually pick ones that are, it depends how we're synthesizing it. If we're synthesizing the peptide by solid phase peptide synthesis, we generally don't want the amino acid length to be too long. We also want it to dissolve well in water, so we check for its pI. But also, what's really important is how the cysteine frameworks are in our, what we find in our transcriptome database. So the cysteine framework is how can I explain this? The cysteine framework, so depending on how the peptide is folded, they're cysteine rich, so they often have these sulfide bonds, so they're stable. And based off the way they fold, they often act similarly to each other. So there's different classes, I believe, like over 25 different classes of cysteine frameworks. So we tried to have a broad range of different cysteine frameworks to see if, one, do they act as they say they're supposed to, and two, so if we have a cysteine framework that targets calcium channels, will it actually target the calcium channels?
Okay, thank you. That's time. So we'll pass it back to Charlotte for the next speaker.
Great. Thank you so much for joining us, Tanya. So
Thank you so much for having me.
we will hand over to our final speaker of our Halloween sessions,